Okay, I'm going to focus mostly on homologous recombination. As I said, this is a situation where the double-strand breaks have occurred, and the end of the double-strand break has to be able to find a partner in order to be able to carry out this repair. In essence, the way this is going to work is that the end of the DNA molecule is going to be used to search other DNA molecules to find locations in which essentially the same sequence has been located. And that can be done by the fact that the end of the DNA molecule is essentially able to make base pairs with the template. Since it has the same sequence, it can make equivalent base pairs to what the original DNA molecule had. And this is the mechanism by which homology can be recognized. And I'm going to talk also about how that actually can be seen. Okay. The mechanism I'm going to start with is called break-induced replication. And it is really the mechanism that we have been sort of focusing on. We saw that stalled replication forks can lead to DNA breakage. The reason that that may be advantageous to cells is it turns out to be a way to restart DNA replication in a recombination-dependent fashion. And this recombination-dependent mechanism we call break-induced replication. Now, I showed you that DNA molecules stall in replication at the presence of photodimers and said that these can be broken. But it turns out that the situation is actually a little bit more complicated than that. The stalled replication fork can actually undergo a little ballet in which the strands of the DNA molecule are rearranged in what is now known as a Holliday junction. This four-way structure was in fact postulated in 1964 by Robin Holliday, who was studying DNA repair, and he suggested that this junction would have unusual properties that would be important in recombination, and of course he was right. So here what's happened is that the two light blue strands, which are the newly replicated strands, are being pulled apart and joined to each other to form a duplex molecule. And the purpose of doing this in part is to push the DNA replication fork back away from the UV photodimer so that this region of the DNA is exposed, and there are DNA repair enzymes that can get at this photodimer and repair it. But in the process of this, one forms this remarkable Holliday junction. We know that this happens because, at least with budding yeast, we can arrest cells in replication using a drug called hydroxyurea. And if one takes electron micrographs of the DNA from those cells, one can actually see these regressed replication forks, these Holliday junctions. Sometimes these structures, as it's noted here, are called chicken feet because of the structures that they resemble. And this has all happened because the replication machinery has arrived at these places and then undergone this reversal of the replication fork, involving also the dissociation of the DNA replication polymerases so that one can end up with this structure. Here's another picture of this structure, which just illustrates the fact that this is a very symmetric structure. The four arms that are generated are in fact symmetrical with respect to each other, and as illustrated here, all of the base pairs of this structure can be made, so that we have four arms of this structure all joined at this common point. This is just another picture which again illustrates that all the base pairs can be formed.